Okay, in this video, we're going to talk about the scientific method, the language of science. Now, it's not a strict like this step to this step to this step because you can kind of move around. It's in flux. It's a process. Okay, but a lot of the parts are the same as you may have learned before. It's just important to understand that it's not simply no 1 through 7 and that's just the way it goes. All right, so here are the objectives for this lesson. Again, you can pause it and read it more carefully if you desire. All right, so what do you know about the scientific method? So again, remember that question mark symbol means this is where you're going to think. So what do you already know? I'm not going to answer because I'm going to answer in this PowerPoint, but think about it. What do you already know? All right, so here's a general flow of the scientific method, which is going to start with some observations, a question like, I wonder. Okay, and then researching. So what has already known about this? And then formulating a hypothesis. Hypothesis, not just a random guess, okay? And then testing it, analyzing it, reporting the conclusions. But again, there can be some flux around, which I'll show you in another diagram in a moment. So it shows the general flow within the scientific method. Hypothesis, remember, is not a random guess. It's based on research. An important part of the scientific method is reporting those conclusions. So it's peer review, it's publishing. And in our classroom, it's lab reports, okay, and presentation. All right, however, the scientific method is not always so neat and linear. Sometimes we change, expand, reject the hypothesis after the experiment and try again. Sometimes the procedure doesn't work and we have to troubleshoot and then revise and try again. Science is about the process. Okay, so it's always going to start though with some observation or question, doing research, and then formulating a hypothesis. And then we're going to design and test, but in the middle of the experiment, you could ask, is the procedure working? If the procedure isn't working, troubleshoot and then go back and start testing. If the procedure is working, then okay, collect your data, analyze your conclusions. And then if your results align with your hypothesis, you're going to communicate those results. And maybe you want to retry it. Okay, if your results don't align or partially don't align, you're still going to communicate those results, but it may lead you back to developing a new hypothesis and designing a totally new experiment. All right, so what is a hypothesis? It's a suggested explanation. It's based on research. It's a testable prediction. So often in science classrooms, you'll find it in this format, if, then, because. So like if plants are placed in darkness, they will not grow because light is required for photosynthesis. So it's not just simply making a random guess. If I put plants in darkness, they won't grow. I need the research piece of it. I need an explanation. Okay, it's a suggested explanation. If an antibiotic is placed on the agar plate, bacteria will not grow because the antibiotic will kill the bacteria. Again, it's saying what you think will happen, but it's providing an explanation as well. Okay. What are the kinds of variables in an experiment? Well, we have the independent variable. That's the cause. That's the variable I, as the scientist, change. And then the dependent variable, that's the effect. That's the variable that changes because of the independent variable. It's the one that is measured, okay? And in a graph, the x-axis is going to be the independent variable. The y-axis is going to be the dependent variable. Okay. Some other important parts of an experiment, we have controlled variables. So variables kept the same to get reliable results. So remember, let's talk about the example, a plant put in darkness. So what would I want to keep the same? Well, I'd want to give the same amount of water, the same amount of soil, the same type of plant, the same age of the plant, okay? The control group, the independent variable, is not changed in this group. So in that case, the control group, they're going to have the light, okay? Experimental group, the independent variable is changed. So I'm putting them in darkness. I'm testing darkness. I put them in darkness. The control group does not have darkness. They would have light. The data that you collect in experiments can be of two main categories, qualitative and quantitative. Qualitative, just like it sounds, is a quality. So we use our senses to notice properties. So color, odor, shape, texture, state of matter, anything that you can observe, but you don't measure it. Okay. Quantitative, just like it says, is a quantity. We use numbers and measurements. So we measure length, temperature, mass, time, speed, any data that can be measured. And then we organize our data, organizing it into tables and graphs. Okay, charts, tables, graphs, very common the way to organize and then describe our data. So how do we describe it? As temperature increases, the pressure increases. So notice that I use the independent variable first and then talk about the dependent variable. So as temperature increases, pressure increases. In the graph above that one, as the time increases, the temperature decreases. And then you can do the same thing with a table. You don't have to only do it from a graph. As the time increases, the temperature increases. Now, these terms are often confusing, so I'm going to try to hopefully make a little bit of sense for you. So there's precision and accuracy and reliability and validity, and they kind of mean the same things and are used interchangeably. So what is reliability and validity? So reliability is really precision. It's how repeatable an experiment is. So it's like you keep getting the same results. Validity is accuracy. It's how correct the results are. So does the experiment actually measure what it says it's going to measure? Does it address the hypothesis? And how can you improve validity and reliability in an experiment? Well, you can repeat the experiment. Well, that gives you some reliability, some precision. You can use a large sample size. Okay, so if I'm doing my plant experiment, I don't use one plant, I use 5 or 10 or 50. I use properly calibrated equipment. So if I am measuring mass, I'm going to use a balance that's calibrated. I use a more precise equipment. So I don't use a ruler that only has centimeter marks. I use a ruler that has centimeter and millimeter marks. Okay, I use a balance that will give me 3 digits after the decimal point instead of just 1. And we control for more variables to help isolate the ones we're studying, the independent and dependent. So control for as many things as you possibly can. Also, using a control group will help. So if an experiment is repeatable, it's reliable or precise. Okay, if an experiment is accurate, it's valid. Okay, it actually measures what it's supposed to measure, and you get close to the actual value. Also, these can be confusing, law versus theory. So a law is just a description of a natural phenomenon. It describes it but doesn't explain. So Newton's law of gravity says that whatever goes up must come down. Law of conservation of mass, mass is never created nor destroyed. Okay, it does not explain the why. It just tells you this is what it is. A theory, though, is an explanation based on evidence, and that evidence acquired through the scientific method, and it's repeatedly tested and confirmed. So the theory of evolution by natural selection, the Big Bang theory, remember, theories explain, laws just tell it like it is. That's it. This is what happens. No why, no explanation. Theory is the explanation. So which of the following definitely indicates an error in an experiment? So take a moment and look. If you violate a scientific law, remember, law is just the way it is. That definitely means you made a mistake. That's totally fine. In class, we just might have time for a blue kit. And there you have it, a quick overview of the scientific method, and hopefully you learned something new.